I'm Charles Npuwe. I'm a scientist at Scion. I work on developing gene editing technology in conifers. Uh, I am new to New Zealand and to Scion. Um, I came from US where I worked on populus species and gene editing in populus for bioenergy. Today I'm going to talk about a brief overview of plant breeding technologies that has been developed over the years, and a little bit about CRISPR and Cas9 gene editing, and then some of the advances we have made uh, in developing sterile trees. Uh, so, humans have been practicing plant breeding for a lot of years, and mostly it has been using traditional breeding where you have uh, two parents with desired qualities, where you cross them and then keep on selecting them till you get your desired phenotype. Uh, in the 1950s, a big change came with the development of artificial mutagenesis. And here the host organism is subjected to radiation or chemicals to induce mutations in the genome. Uh, and these mutations are random. And then again, you select for the desired phenotype. And in the 1990s, the transgenic genetic modification was developed. And here we insert selected genes with known function from species that cannot be crossed into the parent, and then it is inserted into the genome. And it has been used extensively throughout the world. Nearly 90% of the corn in the US is now genetically modified, and there are many countries that grow and use genetic modified crops. And coming to the technology today, we have the most recent one called gene editing technology. So in gene editing technology, we specifically modify a single gene in the genome. It is very precise, it is predictable, and it is one of the safest technology we have so far. So throughout the history, we have moved towards precision in plant breeding. Coming back to traditional breeding, again, as I said before, we have parents with desired qualities which we cross and then subsequently select for the desired phenotype. As you can see on the picture on the left, the smallest one is called the ancestor of the maize. It's called Theosinte, and it has been selectively bred over thousands of years to get to the corn as we know now. And the other example is the different varieties of peppers that has been selectively bred for different colors, sizes, and spiciness. And so we have, most of the domesticated crops were developed through conventional breeding or traditional breeding. And coming to mutagenesis, there are two types of mutagenesis, and one is the natural mutagenesis that occurs in nature, and these are usually caused by radiation or heat or chemicals that are present in the environment. And evolution mainly depends on natural mutations, and it is very slow, and it's rare. One of the best examples of natural mutation is the human eye color, and the other type is the induced mutagenesis, where we use chemicals or radiation, and the host organism is exposed to the radiation to create mutagenesis, and then we select for the desired phenotype. And this causes a lot of random changes in the mutation, but it is also very quick in terms of breeding. And okay, so an example of a natural mutation is, the first one is the blood red orange. And this orange occurred as a single mutation in a branch in an orange orchard. And this was subsequently propagated to be the variety that we know of as the blood red orange. The second example is the semi-dwarf mutant in rice and wheat. And this plant is naturally dwarf, and it produces more tillers. And this was the basis of the Green Revolution that increased fruit production throughout the world. Coming to examples for artificial mutagenesis, so these, as I said before, these mutations are caused by exposing the plant or the source material to chemicals or radiation. So mandarin orange was exposed to gamma radiations, and as a result of that and selecting subsequently, we got the seedless variety of mandarin called the Tango mandarin. This was developed in University of California. And coming to transgenic genetic modification, here the selected genes are selected based on their known function, and then it is inserted into the host genome. And as I said before, it's been used extensively throughout the world. So there are two types of genetic modifications. One is transgenic, and the other one is cisgenics. Transgenics introduces material from other plants that cannot be crossed into the host, parent plant. And cisgenics is also introduction of a gene, but it is from a related species. So this is done mainly to quicken the breeding program. One of the examples of transgenics is the orange petunia that was in the news a few years ago. Naturally, orange petunias do not exist. And this was created by modification of the genome to produce orange petunias. And this had a gene inserted from another species to produce the orange color. And so in the early 60s, American chestnut was almost wiped out by the American chestnut blight. So they started a breeding program to cross chestnut blight-resistant Chinese chestnut into the American chestnut to save the American chestnut. And so after years of selecting and to breed out the Chinese variety, they could achieve only 98% of the American chestnut background. But by using a single gene from Chinese chestnut that is shown to be resistant, cause of the resistance, it was introduced into the American chestnut, and it provides resistance to the chestnut blight to the American resistant, and you get a 99.99% pure background. So as you can see on the insert on the top, on the left is the American chestnut that is susceptible to the chestnut blight, and on the right is the transgenic plants that is resistance to the chestnut blight. And this can be also seen as an example of cisgenics because it is coming from a related species, but it is also considered overall as a transgenic because the gene is from an outside source. Again, every spring in Japan, Japanese cedars produce huge amounts of pollen and thus causes pollen allergy epidemic in Japan. So they created a pollen-free transgenic cedar. And you can see on the insert, on the left is the non-transgenic cedar, and the right is the transgenic cedar. The flower buds or the male flowers look exactly the same, only when it is seen under a microscope, you see no pollen in the transgenic cedar plants. And when you take a cross-section of the pollen sac, you don't see any pollens present. And this trial is being, a restricted trial is being conducted right now in Japan. And coming to the most recent technology in gene editing, and as I said, it is a precise and predictable, and we edit only the gene that is already present in the genome. We are not adding any new genes or new DNA into the plant. And that's why I said it's very precise. We know which region of the gene we are editing, and it is considered a non-GMO in many jurisdictions. And these mutations that are caused by gene editing cannot be detected or easily distinguished from natural mutations. And coming to the CRISPR, CRISPR is the most famous gene editing tool. So these are engineered enzymes that contains two parts. The first part is the DNA targeting part. So that gives the specificity and precision for gene editing. That specific part guides the enzyme to the DNA where it needs to cut. And the enzyme or the nuclease cuts the DNA. And that's how we create mutations. As an example of gene editing to compare it with conventional breeding, Here's shown as an example for tomatoes. If you need to breed resistance or tolerance to salt or abiotic stress, you cross it from different wild relatives, and you finally create a stress-resistant tomato that takes about 10 to 15 years. But through gene editing, we know about 6 genes that can give the very same phenotype. So within 1 to 3 years, you are able to create a gene-edited tomato that is very similar to the one that has been developed through traditional breeding. The only thing is you have shortened the time from 1 to 3 years. Other examples that has been done throughout the world are gene-edited cassava plants to produce more starch, and then the non-browning mushroom in the US that has been edited to prevent it from browning, and then there are cashmere goats that produce more longer fibers, and again the dehorning cattle has been developed in ag research in New Zealand, where you eliminate the production of horns in cattle, so you don't have to dehorn the cattle. And there's been also major push to develop malaria-resistant mosquitoes through gene editing, and it has been demonstrated to work in mosquitoes. And as we know, majority of our trading partners either use or grow GMOs. Japan is one of the largest importers of GMO foods, though labeling is required. They import GM corn, soybeans, papayas, and for flowers they grow roses and carnations, even though they are genetically modified. The countries in the red boxes have said that the gene-edited crops are no longer regulated. So the browning mushrooms, non-browning mushrooms, are not regulated as a GMO in the US and Canada. So this series of pictures that we are all familiar with shows the amount of or the extent of the wilding problem in New Zealand, and as you can see, within a span of 20 years, it has almost covered the whole mountain range, and it has impacted severely the ecology of that region. And one of the programs that I work on is to develop sterile trees to prevent the spread of these wilding conifers. And as we know in other flowering plants, that flowering genes involved in flowering is very complicated, and it also involves various environmental factors. And so we are working on identifying genes that are responsible for cone development, or male and female cone development, or pollen development in conifers. One of the things we have done is the picture on the left shows a blue enzyme that is being targeted to the anthers. So we know which gene can be used to target the anthers, and we also know that gene works in conifers. And so that provides us a step in the direction of making sterile trees. And in order to make sterile trees, and in a non-GMO way, we are working with protoplast, and protoplast are regular cells, and as you can see on the left panel, this is a regular cell where the red border is the cell wall, and we remove the cell wall with a cocktail of enzymes, and then that gives us the protoplast. That's just the cell without the cell wall. And we use protoplast for genetic modification. One of the challenges in using protoplast is to, once you remove the cell wall and you manipulate it, you have to make the cell to produce the cell wall again. And once it produces the cell wall, it has to go on dividing to form a plant. So that's a major challenge for us right now, but we are moving in the right direction. So the general idea is to extract protoplast from trees and then manipulate them and make them grow into trees again. So in Douglas-fir, we have started working on protoplast. As you can see, the leftmost panel is the protoplast that are isolated from Douglas-fir, and we have also made it form the cell wall, and it usually happens within 20 to 24 hours. And in 48 to 60 hours, it starts dividing. And 4 to 6 days, you can see small colonies, and the colonies start producing bigger colonies in about 5 to 7 days. So we are working and towards that production of plants from protoplast, and we are also working on transformation of protoplast to edit the genes and in the Douglas-fir genome. And so we are also trying to working towards, as I said, transforming cells. So here you can see that we have introduced a foreign protein into the cell. So the blue color is caused by an enzyme that we transferred into the cell. So this does not involve in foreign DNA. It just uses the protein, and once the editing is done, and then the protein will degrade and go away. So there's no foreign gene added to it. And besides non-transgenic gene editing, we are also working on transgenic gene editing. To produce sterility in Douglas-fir, we have identified genes in the Douglas-fir genome, and we have analyzed their expression in different parts of the tissue, and we have genes that are very specific to male and female cones. And some of the sterile genes have already been transformed into Douglas-fir. And for gene editing using the transgenic method, we have identified target genes, and they have been sequenced, and they are being used for gene editing. And so we have right now in our greenhouse Douglas-fir that has been predicted to be male-sterile. These are approximately 1 year old, and they're growing in our greenhouse. With that, I would like to thank both our tissue culture and the biotransformation team, and especially to Sanjeev, who did most of the protoplast work, and to Steffi, who did identification of the genes in the Douglas-fir genome. And also I would like to thank the Forest Growers Research for funding this work. Thank you.